Okay, we're going to start chapter 14 today. Last chapter, you guys. Woohoo! Semester's almost over. Chapter 14 is the last biomolecule. We already talked about carbohydrates, we talked about lipids, we talked about proteins, and now we're going to talk about one that's a little different because those other ones are kind of in foodstuffs and things like that. Now we're going to talk about nucleic acids, which are a biomolecule but a little bit different than the other three. Okay? When we talk about nucleic acids, okay, they're made out of basically three things. They're made out of nucleotides, and the nucleotide is made out of three parts. You can see there's a phosphate group, and by phosphate I mean PO4. There's a monosaccharide, okay, so we've got, a carbohydrate here, and then we have a base. So like we talked about before, we said that carbohydrates are made out of monosaccharides, lipids are made out of triglycerides, and we have proteins made out of amino acids. Now we have nucleic acids. They're made out of nucleotides. Okay, so there's three parts to a nucleotide. You have a sugar, and if it's DNA, it's deoxyribose. If it's RNA, it's ribose. That's where their names come from. And again, this is our phosphate group, and this is our nitrogen base. And there's 4 different nitrogen bases in DNA, and then there's 4 in RNA as well, but there's one different, and we'll talk about that in a little bit. Okay, so when we're talking about these two, DNA and RNA, those are our nucleic acids. Here's the sugar, the monosaccharide. Okay, the only difference really is if you look at deoxyribose, it has just a hydrogen here, where ribose has an OH group here. So deoxy, one less oxygen. Okay, so this is the sugar in DNA, this is the sugar in RNA. Okay, and when they talk about counting, they go, the prime number is here, it starts at this one and it goes around. So it's 1, 2, 3, 4, 5. Same here, 1, 2, 3, 4, 5. Okay. All right, so when we look at nucleotides, again, they contain a nitrogen base, and there's 2 different groups. There's purines and there's pyrimidines, okay? If you look at them, see, here's the purines, which is adenine and guanine. These are 2-ring carbons. And over here, the pyrimidines are 1-ring, okay? And the reason why there's 3 of these and only 2 of these, when you look at DNA, in DNA, adenine, this one, always goes with thymine, okay? So a 2 goes with a 1. And then in RNA, it's different. Adenine goes with uracil. DNA doesn't have uracil, and RNA doesn't have thymine. So these 2 guys, and again, the only difference is thymine has a methyl group here, and uracil doesn't. It has just a hydrogen. But again, you always have a 2-ring with a 1-ring. So adenine always goes with thymine in DNA. And over here we have guanine, which is a 2-ringer, which is called a purine, and it goes with cytosine. That's a pyrimidine. And by a 2 going with a 1, it keeps the middle part of your DNA where your nitrogen bases are, then they're always the same distance. Okay, if you put a 2 with another 2, these guys don't match together. Okay, then you'd have a wide and then a narrow, and a wide and narrow. Instead, they're always the same distance because, again, a 2 goes with a 1. Guanine and cytosine always go together. Adenine and thymine always go together. And if you're talking about RNA instead, adenine and uracil would go together. So the G and the C are always the same in both. The only difference is RNA doesn't have thymine, it has uracil. Or DNA doesn't have uracil, it has thymine. But when you're matching up your nitrogen bases, they always go, the order is G and C always go together, and A and T, if you're talking DNA, always go together, or A and U always go together if you're talking about RNA. That always is the same. Those always match up, okay? If guanine tries to match up with thymine, it's not going to happen, okay? Okay, so the nucleotides, remember they have a sugar, we talked about, and the nitrogen bases, and then they have a phosphate group, okay? It's part of what they call the backbone. So the backbone, the size of DNA is sugar, phosphate, sugar, phosphate, sugar, phosphate. In from the sugar is your nitrogen base. And both sides are the same. The backbones are both the same. Sugar, phosphate, okay? It has the same name. The nucleotide has the same name as what they call the addition of the number 5 phosphate, okay? So nucleotides, again, are derivatives of phosphoric acid, okay? That's where the deoxyribose nucleic acid comes from. They're a derivative of that. That's where it comes from, the phosphate group. Because to get the phosphate group, you have to have, it comes from phosphoric acid, and that's another part of the name. Who would have thought that so much was in this name? Actually, when I was in college, we had to give a speech on one word. That's kind of hard, but I use deoxyribose nucleic acid because that's a nice long, and then my speech was fine because I had lots of information. Sorry, had to give you that. Nucleotides, there's also what they call NTPs. They're common energy forms. If you remember from biology, if you've taken biology, when they talk about ATP, adrenaline triphosphate, that's actually a nucleotide. It has adenine and it has, the sugar base and whatever, but its phosphate groups can come off and reattach. So the phosphate is where the energy is. Okay, so we have one other kind of nucleotide. ATP would be the most common, and ADP. Okay, that doesn't have anything to do with DNA and RNA. Okay, so but there is more to nucleic acids than just DNA and RNA. Okay, so if you're looking at the structure, the chemical structure of RNA and DNA, they're called linear polynucleotides. In other words, they're long chains, and it's a bunch of nucleotides together. Okay, they go from one side, which is the number 3 hydroxyl group. Remember, we numbered them 1, 2, 3. So they go from the number 3 to the number 5, and the number 5 would be over here. So when they start, they go number 3 to number 5, and then on the other side it would number 5 to number 3. Okay, and again, that's just the number of the carbon. Okay, so again, here's my sugar, here's my phosphate group, and then I have a nitrogen base. So then in between I have another phosphate group that attached to this sugar, and then we have another nitrogen base. Okay, so on this side, see, my nitrogen base is A, so then when it comes in from the other side, T is going to come in here, and here I have C, then G is going to come here, and so on. They're going to match up with their counterpart. Okay, so when you're writing the polynucleotide, again, to list the nucleotides, they start from the 5 to the 3 end, and then the other side is the 3 to the 5 end. Okay, the reaction that joins a nucleotide to the chain is actually an alcohol and a phosphate. They form a phosphate ester. Not a huge deal, but I'm hoping you remember what those words mean. It's just the C double bond OO. Okay, that's going to form that group. But again, we have all these organic reactions that are happening, and they happen in DNA to form this long, super long chain. Because if you think about DNA, it's made out of millions and millions and millions of different nucleotides. Well, how do they join together? Okay, they go from the number 5 prime, that number, the carbon, go down, and then the other side would be the number 3. So they're opposite on each other, and then they match up in the middle. If A is sticking out, then T is going to come in. If C is sticking out, G is going to come in. They're going to match up with their nitrogen base, with their partner. Okay, they do need some energy, so ATP is going to be used. Okay, and again, the reaction occurs from the 5-phosphate to the 3-hydroxyl, and then they grow. And on the other side, it's going to be just the opposite. It's going to go from the 3-hydroxyl to the 5-phosphate. So that you'll see sometimes on there, they'll have 5 to 3 to 3 to 5. And again, that's just which carbon they're on. Not a huge deal, you guys. It's more important that you understand that the A goes with T and C goes with G, and that never changes. Okay? Okay, I think, oh yeah, we'll do this one. DNA is made out of 2 polynucleotides. In other words, 2 strands. You got phosphate sugar with the nitrogen bases sticking inward, and then the same thing on this side. Okay, nitrogen, you got your bases, and they join together. Okay, and then eventually what they're going to do is they're going to twist. And they've discovered the twisting, the helix, and I'm sure you've heard of DNA as a double helix. It's like a spiral staircase, but it twists right-handed, and the twist, they think, holds it together better. When it's straight ladder with all the steps in there, if it stayed that way, it's not as stable as if you just twist it. The twisting keeps it together better. Okay, so again, the sides, if you're thinking of DNA as a ladder, the sides of your ladder are sugar phosphate, and they just keep going. And the inner part of, or your steps of your ladder, that's your nitrogen bases. Okay, and again, they're called anti-parallel because they move in opposite directions. Okay, you'll be watching a video and it'll explain that probably better than I just did. Okay, and how did they figure that out? Well, that's kind of a mystery because if you look, the people that get credit for discovering DNA structure is Watson and Crick. Um, James Watson was at U.S. Francis Crick is from the U.K. and they worked together and they figured out the reason and so on and so forth. Well, in order to get their results, they had to have an X-ray data picture from Rosalind Franklin. The thing is, she didn't give it to him. It was taken, her work was basically taken from her by her supervisor and given it to these guys. So they use it to to affirm what they thought. And so when it comes to the structure of DNA, it should be Watson, Crick, and Franklin. And she should be first because without her work, they would have never got their work done. Unfortunately, by the time they discovered all this and they're ready to hand out the Nobel Prize, Rosalind Franklin had passed away. So she never really gets the credit she deserved. But so Watson and Crick are the one that are always give credit for discovering it, but it should be Franklin, Watson, and Crick. That's my feminist movement today, you guys. Without her work, they never would have figured it out. Okay, so there's our DNA. Okay, we have the base pairs in the middle. We have the sides, these like ribbon-like structures. Those are your sugar, phosphate, and they just go back and forth, back and forth, back and forth. The nitrogen base always comes from the sugar. It's not attached to the phosphate base. So that's a nucleotide. If you think about a bunch of Legos, each individual Lego would be a nucleotide, and when you put a whole bunch of them together, then you got your DNA. And that's probably a bad analogy, but anyway, they just keep snapping together, and then you get this code in the middle. And this code is what's really important about DNA, because this code is what determines what protein you have. Okay? This code is unique to you unless you're an identical twin. Okay? Nobody has the same DNA as you. And again, that's a big thing for forensics, for crime too, because once they discovered that, it was like, hey, that is your DNA. We're 99.9% sure that's your DNA. Unless you have an identical twin, that's your DNA. So anyway, that's the structure of DNA. I'm sure some of you guys have already gone through this in other classes. It's a review, but DNA is one of the nucleic acids. It's a biomolecule. Okay? So we'll quit there for today.